Today, we will talk about nutrigenomics, customizing your diet based on DNA. Nutrigenomics is the science of how your food talks to your genes. It studies how what we eat can switch certain parts of our genes on or off, helping us avoid or treat diseases. Nutrigenomics makes it clear that everyone is unique and can react differently to the same food due to their individual genetic makeup. This means we can use nutrigenomics to personalize our diets for better health. Nutrigenomics offers a way to prevent or manage chronic diseases like obesity, diabetes, heart disease, and cancer. These are conditions that are influenced by both our genes and our environment, including the food we eat. Nutrigenomics can also help us find new ways to measure disease risk and progression, and even identify new potential drug targets. In this video, we will take a look at the basic principles of nutrigenomics, such as the two-way interaction between genes and nutrition, and highlight the practical uses and challenges of this exciting new field. The nutritional genetics, a closer look. Think of genes as small bits of DNA that contain recipes for making proteins, which are the essential building blocks of life. Proteins have many functions in our cells, like speeding up chemical reactions, moving molecules around, controlling gene behavior, and sending signals. Tiny differences in these genes among individuals lead to different versions of proteins that can affect our health in various ways. Nutrition is about getting and using food for energy, growth, repair, and maintenance of our bodies. It can impact how our genes behave by affecting the supply of molecules required for making, changing, and repairing DNA, such as vitamins, minerals, antioxidants, and certain chemical groups. It can also influence gene behavior by changing the activity of enzymes that control how genes work. The relationship between genes and nutrition is a two-way street. Genes can determine how we break down and react to nutrients, and nutrients can influence how our genes behave. For example, some people might have a gene variation that lets them convert beta-carotene, which the body can change into vitamin A, into its active form more efficiently. This means that these people might need less beta-carotene in their nutrigenomics diet to get enough vitamin A than others. Conversely, some nutrients can change the behavior of genes linked to inflammation, oxidative stress, cell regulation, and cell death, which can affect disease prevention and treatment. For instance, omega-3 fatty acids can lower the activity of genes that cause inflammation and boost the activity of genes that defend against oxidative damage. These are just some nutrigenomics examples that show how a nutrigenomic testing and subsequent diet plan can greatly impact our health. Simplifying nutrigenomics. Nutrigenomics seeks to understand how different types of food and nutrients can change the way our genes behave, and how our unique genetic variations can alter the way we react to different diets. The ultimate aim of nutrigenomics is to help us find out how we can prevent or treat illnesses by adjusting our diet to match our unique genetic characteristics. Nutrigenomics is also known as nutritional genetics, a term that covers various related fields such as nutrigenetics, nutrigenomics, and nutritional epigenetics. Each of these fields help us understand different ways our genes respond to nutrients and show certain traits like risk of illness. Nutrigenetics looks into how different genes can influence the way our bodies react to our diet and the risk of getting certain diseases. For instance, some individuals may require more or less of certain vitamins or minerals based on their genetic makeup. Nutrigenomics, on the other hand, explores how our diet can change the way our genes work. Some types of food or nutrients may turn certain genes on or off, controlling things like blood sugar levels or the production of cholesterol. Nutritional epigenetics is the study of how diet can change the chemical tags on our DNA that control gene activity without altering the DNA itself. For example, certain foods or nutrients may influence how our DNA is modified, effectively turning genes on or off. Nutrigenomics is an area that brings together many scientific fields like nutrition, genetics, biochemistry, molecular biology, physiology, and bioinformatics. It uses advanced tools that can process and analyze large amounts of data, looking at various levels of biological information, such as genomics, transcriptomics, proteomics, and metabolomics. Genomics is the study of the full set of DNA in an organism. It can help identify genetic differences that could change the way we react to certain nutrients or our risk of getting diseases. Transcriptomics looks at all the RNA molecules in a cell or tissue, which can give us insight into how genes are behaving under different nutritional conditions or states of disease. Proteomics is the study of all the proteins in a cell or tissue. This can give us insights into the structure and behavior of proteins involved in different processes in our body. Metabolomics studies all the small molecules in a cell or tissue. This can help us understand changes in our body's chemical reactions due to diet or diseases. Nutrigenomics gives us a deeper understanding of how our diet and genes work together to affect our health. Here are some examples. On a molecular level, Nutrigenomics can show us how specific foods or nutrients can influence the way genes work. For example, curcumin, a compound found in turmeric, can control the genes involved in inflammation and cancer. At a cellular level, Nutrigenomics can tell us how certain foods or nutrients can change cell functions. For instance, resveratrol, a compound found in red wine, can activate certain proteins that control metabolism and lifespan. On a tissue level, Nutrigenomics can show us how certain foods or nutrients can influence the balance of tissues. For example, lycopene, a compound found in tomatoes, can slow down the formation of new blood vessels that support tumor growth. At an organ level, Nutrigenomics can explain how certain foods or nutrients can affect how organs function. For instance, capsaicin, a compound found in chili peppers, can stimulate the release of a hormone that regulates sugar levels and appetite. On a system level, Nutrigenomics can reveal how certain foods or nutrients can influence responses across our entire body. For instance, quercetin, a compound found in apples, can control the immune system by reducing inflammation and oxidative stress. Think of personalized nutrition as a diet plan designed specifically for you, taking into consideration your unique body needs and tastes. This individualized food plan considers aspects like your genes, the way you live, your health condition, and the kind of environment you're exposed to. By focusing on these factors, personalized nutrition could help you achieve better health and fight or manage long-term diseases. Nutrigenomics can tell us why people react differently to the same food or nutrient based on their genetic code. To give some Nutrigenomics examples, some people may have genes that change the way they break down certain nutrients, like folate, caffeine, or omega-3 fatty acids. It can also explain how compounds in our diet can control genes that are part of various biological functions, like inflammation, oxidative stress, or detoxification. What do you think of our video? Let me know in the comment section below. Before you go, please hit the like button and subscribe to my channel. Thanks for watching.